Antibody-drug conjugates, or ADCs, have revolutionized cancer therapy, but their evolution is far from over. The future of ADCs lies in smarter conjugation chemistry, moving away from the traditional one-size-fits-all approach towards site-specific conjugation that enables next-generation ADCs with unprecedented precision and control. This report will explore the conjugation chemistry trends reshaping the ADC landscape through 2028 and the strategic IP implications for innovators and investors alike. This report is based on our proprietary 𝗬𝗧𝗧𝗚𝗏 ADC database, which contains detailed information on more than 1,000 ADC products in development and on the market. Section 1, our analysis reveals 3 key platform technologies, ConjuAll, GlycoConnect, and SMARTag, are driving the transition to site-specific conjugation. With a total of 500 disclosed programs, these platforms offer a compelling risk-adjusted path to developing safer and more effective ADCs. Notably, ConjuAll takes the lead with 43 disclosed programs, including 5 in clinical development. GlycoConnect and SMARTag follow closely with 15 and 12 programs respectively. Looking at the broader pipeline, we see that of the 445 pipeline assets using site-specific conjugation, those utilizing leading platforms demonstrate a 2x higher clinical progression rate compared to non-site-specific ADCs. Furthermore, they exhibit improved pharmacokinetic profiles, fewer off-target effects, and other benefits consistent with greater control over the conjugation process. Section 2, site-specific conjugation is not just a growing trend, it's becoming the standard. In 2024 alone, 57 site-specific ADCs were added to the pipeline, surpassing all previous years. And the momentum is accelerating. Site-specific platforms now represent approximately 67% of all new ADCs in 2024, up from approximately 30% in 2020. This growth is statistically significant and correlates with increased regulatory confidence in homogeneous bioconjugates. Section 3, several technological trends are shaping the field, each presenting both opportunities and challenges. First, there's an evolution in DAR strategies. High DAR ADCs is greater than or equal to 5 have grown from 13% of all ADCs in 2020 to 46% in 2024. Second, there's a clear move beyond Maleimide chemistry. Maleimide-based conjugation is reaching saturation mainly due to inherent instability and aggregation risks. As a result, emerging chemistries like click chemistry, enzymatic conjugation, and orthogonal linkers are gaining traction. These methods offer higher specificity, greater modularity, and can accommodate diverse payloads. Notably, click chemistry approaches like those from Argenx and Qimmib utilize stable non-covalent interactions such as Diels-Alder reactions for conjugation. Meanwhile, companies like Biopharma and Antibody Solutions are harnessing enzymatic conjugation, while Server and others are exploring orthogonal linkers. Third, there's an expanding payload and conjugate diversity. New strategies like dual payload ADCs and degrader antibody conjugates, or DACs, are being explored. These innovative formats, along with oligonucleotide antibody conjugates, require flexible, modular chemistries that can support complex structures. Platforms like GlycoConnect and SMARTag are facilitating this by enabling branched or mixed conjugation. Section 4, a critical challenge in ADC development is identifying the optimal conjugation site S on the antibody. The impact of position on conjugation chemistry is increasingly recognized, with factors like local hydrophobicity, steric accessibility, and payload compatibility playing crucial roles. Notably, a recent study by Genmab highlighted how subtle changes in amino acid sequence can influence site performance. As a result, tailored conjugation site screening is becoming a preclinical essential, impacting both potency and manufacturability. Section 5, assessing the IP landscape, reveals strong positions around conjugation chemistries, particularly enzymatic and click-based systems. However, opportunities remain. Freedom to operate, or FTO, evaluations indicate clear paths for emerging players. For instance, FTO is favorable for those looking to license proven platforms like SMARTag. Similarly, there's potential for new entrants to file patents on novel linkers, especially those compatible with high DARs, as well as orthogonal chemistries. Section 6, we predict 5 key trends through 2025 to 2028. First, site-specific ADCs will dominate. Risk-adjusted probability, 90%. We expect site-specific platforms to comprise more than 75% of new INDs by 2027. This prediction is driven by the clinical success, patent protection, and regulatory alignment favoring homogeneous bioconjugates. Second, there will be a shift toward high DAR greater than 6 therapeutics. Risk-adjusted probability, 80%. High DAR ADCs will likely constitute the majority of late-stage candidates by 2027, supported by linker innovations and validated efficacy. Third, we'll see widespread replacement of Maleimide chemistries. Risk-adjusted probability, 75%. Maleimide linkers will likely phase out as click. Enzymatic and orthogonal strategies offer superior stability and control. Fourth, platform licensing and IP consolidation are likely. Risk-adjusted probability, 70%. Expect biotech and pharma to increasingly license established conjugation platforms to reduce development risk and accelerate timelines. Fifth, new modalities will emerge. Risk-adjusted probability, 60%. Non-traditional conjugates like DACs and oligo-ADCs will likely enter the clinic, demanding flexible, modular chemistries and redefining platform capabilities. Conclusion: ADC conjugation chemistry is evolving into a decisive competitive advantage for next-generation biologics. Companies investing early in site-specific platforms with strong IP protection, strategic partnerships, and robust site screening capabilities will be well-positioned to lead in this dynamic market. As the field matures, expect continued innovation, platform consolidation, and a renewed focus on developing safer, more effective ADCs for patients. Thanks for watching.